The term DNA stands for deoxyribonucleic acid. And when we look at that chemical name, it does tell us a bit about the structure of the DNA molecule. So first of all, DNA is a polymer. And as a polymer, it is going to be made up of smaller units. And those smaller units or monomers are going to be called nucleotides. So with DNA, it's actually a very, very large polymer. And if we were talking about the human genome, and if we just talk about the haploid human genome, we have 3.8 billion nucleotides. And I'm just going to abbreviate that as NT. So there will be a whole lot of these nucleotides joined together. And when we're just talking about the scale of a single chromosome, we're usually talking about a couple 100 million nucleotides linked together. So let's talk a little bit more about that nucleotide structure and what the different parts of it are. So within the nucleotide, there is going to be a phosphate group. We will also have the sugar component, and for DNA, that sugar is always going to be deoxyribose, which is where the D in the name comes from. And then there's also going to be a base component. So if we were to draw all of that in front of you, we would have the phosphate, and the phosphate, recall, this would be the overall structure of a phosphate right here. Attached to that phosphate group shown in green, we're going to get the sugar component. And the sugar component, it is going to be deoxyribose. And deoxyribose is a pentose sugar, which means it's a 5-carbon sugar. So this sugar component, it does have 5 carbons. And we are, since it's deoxyribose, we're missing the hydroxyl group that would be attached right here to this particular position. So that will be the difference between what we're calling deoxyribose and ribose, which we get when we start looking at RNA. Now, if we go on and we talk about the third component, the third component is going to be the base, and the base is going to get attached to the sugar. It would be attached over here. And so I'm just going to draw it out like this and just write base, because the base is really a variable component. We'll have different options that can be in that particular position. If we talk a little bit more about the nucleotide itself, if we number the carbons in the sugar, so that's number 1, this is 2, 3, 4, and this would be the number 5 one right here. We do very often refer to the 5' end of a nucleotide or the 5' end of DNA, and then we also talk about the 3' end quite a bit. Well, if you look down here, first of all, at where the 5' carbon is, the 5' carbon is always, if you just look right here, the 5' carbon is always going to be attached to a phosphate group. So we want to start thinking about the 5' end of the nucleotide or the DNA molecule as the phosphate end. And then if we look at where the 3' carbon is, the 3' carbon is always going to be attached to the hydroxyl group. So we want to think about the 3' end as the sugar end, or in other words, that's going to have the hydroxyl group on it. And those terms are going to become important when we start to link these nucleotides together to make the long polymer of DNA. So this is the beginnings of DNA structure. If we talk a little bit more about those bases that we would have, so what I've drawn here in purple, we are going to have different options for that particular position. Here we can see the 4 bases that are found in DNA. These bases a lot of times are called nitrogenous bases, and that term nitrogenous base just refers to the fact that they do contain nitrogen inside of them, and these bases do fall into 2 major categories. So the A and the G, or what we call the A and the G, so we do refer to these by just the first letter in their name, these 2 are going to be what we call purines. And the purines, they are the larger of the 4 bases. And these are going to be doubled ring structures, as you can see here. So they're going to have a 5-membered ring connected to a 6-membered ring. And then the other 2 bases that we see on here, we have thymine, which we call T, and we have cytosine, which we call C. These 2 are going to be of the type called pyrimidine. And the pyrimidine bases are obviously going to be the smaller ones. Those are just composed of a single ring each. Now these 4 bases that we have, they will be very important to the overall structure of the double-stranded DNA molecule because they are going to be interacting with each other. So we will come back to these 4 bases when we start talking about how the chains of DNA actually connect and hold together in that overall double helix structure. So what we're seeing right here is what happens when all of these nucleotides are connected together. So the nucleotides themselves are going to get connected by bonds that we call phosphodiester bonds or phosphodiester linkages. And these phosphodiester linkages are going to take place, and I'll just kind of circle it on here, they're going to occur between the 5' phosphate of one nucleotide and the 3' hydroxyl group of the next nucleotide. So that's what I've tried to circle right here. So here we can see this would be a phosphodiester linkage, this would be one down here, and this would be one right here. So now I have circled all of the phosphodiester linkages in this strand. And if we just wanted to kind of highlight them a little bit more, specifically they would be these linkages that I'm showing in blue here. Okay, so those are going to be what connects these monomers together to give us this very long polymer chain. Once we have done that, what's going to happen is that that molecule now has a unique orientation or a unique polarity to it. And what we mean by that is notice that one end is going to have just the 5' phosphate group on it. And then the other end down here is where we would find the 3' hydroxyl of the sugar. So strands of DNA are always going to have a 5' end and a 3' end, really regardless of how long this molecule actually happens to be. Now the other side of this we haven't talked about, but notice you have this other very similar side, and that is because DNA is double-stranded. And when we refer to it being double-stranded, that means that there are 2 chains of DNA that are actually connected together. So here we can see that other chain, we still have phosphodiester linkages, which I can highlight again here, that are holding those individual nucleotides together. And if we look at the ends of this particular chain, this would be the 5' phosphate end down here at the bottom of this one. And this would be the 3' hydroxyl end up here at the top. So one thing to really point out here is notice that the strands are running in opposite directions. And this is always going to be the case when we have 2 DNA strands together, or in other words, when the DNA is double-stranded. And we refer to this property as the strands being anti-parallel to each other. Again, so this is really referring to the fact that one of them is running this way and the other one is running in the opposite orientation. Other things to notice about this structure. It is the sugars and the phosphate groups that are on the outside of this overall large molecule. And on the inside, these very colorful components, those are the bases. So the nitrogenous bases that we looked at before. So on the inside, we can see the bases from each of the 2 strands. And these bases are going to interact with each other, and we're seeing that as little black lines here. They are what is responsible for really holding the 2 strands together. Those interactions that we're seeing, those are hydrogen bonds. So that's written right here, but I'll write it a little bit bigger. Those are our hydrogen bonds. And hydrogen bonds are really only going to form between specific base pairs. That's the way that we refer to it. So notice here that we have an A is always across from a T. And the reason for that is because A and T are able to form 2 hydrogen bonds. So I'm going to draw that as 2 little dashes right here. That will always be the case. So wherever you see an A or an adenine in one of our DNA strands, there will be a thymine in the DNA strand that's directly across from it. The other base pair is going to be C and G, and they happen to form 3 hydrogen bonds between them. And really that's the only way that these bases are going to connect with each other. So these combinations, the A and the T and the C and the G, we do refer to those as the base pairs. Okay, and they will have lots of hydrogen bonds between them when you consider that we've got millions of these nucleotides linked together. And that's going to result in a very strong interaction that's holding these two strands together. So that's the overall structure of a double-stranded DNA molecule, which is the way that we tend to see them inside of any cell or anywhere that we have DNA, because these base pairs really want to stick together by forming those hydrogen bonds. The only other aspect to this that we haven't mentioned is the fact that DNA is a double helix, and since it's a double helix, the strands are going to be just a little bit twisted so that we do have an overall rotating or helical structure to this molecule.